Assalamu alaikum. Biomedical engineers create semi-living cyborg cells that could revolutionize health environment. These cyborg cells may be used to create novel drugs and clean up pollution, according to the researchers. A living replica of Dutch painter Vincent van Gogh's famously severed ear is displayed at culture and media museums at KM in Karlsruhe, southwestern Germany on 4 2014. The ear is part of the exhibition Suga Bay with Diamond Streep, an artist specialist in artworks using biological material who collaborated with Bay scientists to reconstruct the Dutch master's ear using DNA from a relative and 3D printers. University of California biomedical engineers have developed cyborg cells which are semi-living but cannot reproduce. However, these synthetic cells and could have large applications, especially in creating novel drugs and cleaning up pollution, according to the university's press release on January 18. Cells with new functions. The cyborg cells were made possible by synthetic biology, which primarily aims to create cells that can carry out new functions, according to Chimeng Tan, senior author of the study and associate professor of biomedical engineering at UC Davis. There are essentially two methods of synthetic biology. One of the to take a bacterial cell that is already alive and modify its DNA by new genes that give it new capabilities. The alternative is build an artificial cell which scratch using biomolecules and a synthetic membrane. The first method uses an engineered lived cell which is very flexible but capable of self reproduction. However, this may not be ideal. A complete artificial cell is incapable of procreation and has a narrow range of functions, but on the brighter side, it is less complex. Third approach, Tan and the UC Davis group then developed a third approach. They injected the building blocks of an artificial polymer into living bacterial cells. Once within the cell, UV light exposure caused the polymer to cross-link into a hydrogel matrix. All of the cells could continue to function biologically. They were unable to divide. According to Tan, the cyborg cells can be programmed, maintain necessary cellular functions, and acquire non-native abilities without deriving. The research team discovered that the cyborg cells were more resilient to stresses that would normally kill normal cells, such as contact with hydrogen peroxide, antibiotics, or high pH. In the end, they were able to modify the cells so that they could infiltrate lab grown cancer cells. The team is inducing an additional study on the creation and control of cyborg cells as well as the effects of various matrix materials. Additionally, they want to investigate how they may be used for a variety of purposes, from addressing environmental issues to identifying and treating diseases. Finally, we are interested in bioethics of applying cyborg cells, as they are cell-derived biomaterials that are neither cells nor materials than said in the statement. Thanks for watching my video. I love it.